In the 1950s, it seemed like psychoanalysis would serve two functions. One, it would give you a penetrating insight into the human mind, the nature of motivation, the nature of action, irrational behavior. And I was particularly interested because of my Viennese background. I'm Jewish. I was born in Vienna. I was in Vienna when Hitler came into Austria. And I saw the Viennese convert from being, civilized, friendly human beings who read Goethe and Schiller and listened to Beethoven and Mozart, and all of a sudden beat up on Jews the very next day. And how can people flip like this? So at first I studied intellectual history, and then I got fascinated by psychoanalysis. I thought it more appropriate to give me insight into mind. And I went to medical school with the specific purpose of becoming a psychoanalyst. And in my senior year at NYU Medical School, I graduated from Harvard College, I went to NYU Medical School, I decided that even a psychoanalyst should know something about the brain. At that time, brain science was a very small discipline. No one at NYU was working on it. There was a terrific guy here at Columbia, Harry Grunfest. So one of my early contacts with Columbia was coming here for this 6-month elective period. And I fell in love with research. I had no prior interest in science. I mean, I took the science courses and I took the first couple of years of medical school, but I loved medicine because of the clinical experience and because I thought I was preparing myself to become a psychoanalytically oriented psychiatrist. But the idea of working with your own hands, setting up your own experiments, thinking with supervision about what the next day's experiments would be about was just exhilarating. And I got a wonderful initial feeling about Columbia from being here. And that experience altered my life. So when I went to see Grunfest, he said, "What do you want to work on?" And I said, "I would like to work on the anatomical localization of the ids, the ego, and the superego." And his eyes glazed over. He thought I was a nut. He said, "What are you talking about? We don't know whether these concepts have any validity. And even if they did, we just don't have the techniques for studying this." So to realize how far brain science was from coming to grips with meaningful psychological problems, that distance was obscure to me. I was not aware of it. In fact, I must say, if I step back and ask myself, look, you've been doing science for over 50 years, what is the transformative change in the field? What is really the dramatic evolution of the field? What is the most important thing? I would say it's the coming together of psychology on the one hand, neuroscience on the other, with sort of an energizing force from molecular biology. That analysis has, throughout my career, even though I sort of gave it up, been a motivating factor in my life. I want to understand how the human mind works. I've worked on simple organisms, but I'm not interested in the snail as a snail. I'm interested in the snail as a model system for understanding how you work and how I work. Since I didn't come from a scientific background, I felt quite insecure the first 8, 9 years I was doing science, whether I could make a go of it. I did a very beautiful set of experiments that drew a lot of attention to themselves. And I thought, beginner's luck. You know, 2 guys, another young colleague who joined me after a while, and myself, Alden Spencer, and I were lucky. You know, we hit a triple the first time at bat. I then took a residency in psychiatry after I left the NIH. I did another project. So the first project was to study the hippocampus, the area known in people to be involved in memory storage. And Alden and I were the first people to do cell biology of hippocampus. And while I was doing that, I realized this is awfully complicated. If I want to understand memory, I can't start with anything this complicated. I've got to go to a much simpler case. And I began to focus in on the snail, and I selected the snail to work on. And by that time, I'd already been married to Denise for a couple of years. And she came from France. We went back to France. The only 2 people working on it were in France. And I committed myself that in 1962, after 2 years of residency, I would go back to full-time research, work on the snail. But while I was a resident, my chairman was extremely supportive of me, and I did research. And I applied the same techniques that I used in the hippocampus to study the hypothalamus. I asked, do cells that release hormones, they're called neuroendocrine cells, have the same properties of conventional nerve cells? And I showed that. Very nice demonstration. I said, very nice. Aren't you lucky you did it the second time, but you applied the same thinking, the same logic to another structure. There's no fundamental idea here. This is not great creativity. And it wasn't until I got going on the snail and I see that a key part of research is simply showing up every day. That, you know, the data will speak to you, and at first, of course, one works by oneself, so it's essentially your own ideas or those that you get by exchanging information with some senior people. But then after a while, you've got people in your lab, and they've got lots of ideas, and, you know, the important thing of doing a collaborative set of experiments is not whose idea it is, but what are the good ideas, and let's run with them. So after, you know, several years of doing things that were interesting, I said, you know, maybe I can do this. And I gave up all, I would say, 1965, 66, I gave up all plans of becoming, of combining the careers or doing psychiatry seriously. It actually came at a significant crisis in my life. So in 1964, 65, I was offered at a very young age, it must have been about 35, a chairmanship of psychiatry at Harvard. Well, my God, I would have killed for that while I was in medical school. By the time that offer came to me, I was really so committed to science. My wife said, you're going to throw your career away to take an administrative job? That's crazy. And I completely agreed with that. And since then, you know, I think about psychoanalysis as sort of a motivating worldview, but I don't think of myself as a psychiatrist. I think of myself, you know, as a working scientist. We were the first ones to show how learning works. The learning involves a change in how nerve cells communicate with one another. And you can strengthen a reflex through learning by strengthening the connections between cells that make up the reflex. And we showed that with short-term memory, memory of the last minutes, that's a functional change. You don't see any change in the anatomy of the connection, in the anatomy of what we call the synapse. But with long-term memory, you see the growth of new synaptic connections. I mean, to see that is just absolutely spectacular. We then began to explore the molecular underpinnings of it. You know, what is responsible for the functional change? What's responsible for the growth? And we found the functional change was due to the activation of a signaling pathway within the cell called the cyclic AMP pathway. Now, this had been described by years before, had been extensively studied even in the nervous system by my colleague Paul Greengard, but no one knew exactly what its physiological function was. So we found a specific function for it, and we found it in a fascinating context. You know, what's more interesting to me than learning and memory? And that encouraged us to study how long-term memory occurs. And we found that the signaling system with long-term memory moves into the nucleus. And there it activates genes. And it's the activation of those genes that gives rise to the growth of new synaptic connections. And we soon identified the nature of the genetic switch. It's a gene regulator called cyclic AMP response element binding protein, CREB. And that switch has turned out to be almost universal. I mean, not just in the snail, but in the fly, in the mouse. Every form of conversion from short-term to long-term memory involves CREB. In a number of important cases, it's the key switch. Essentially, you know, not the only one, but absolutely fundamental. Take it out, everything is lost. In some cases, there are other components of the switch as well. But even now, new studies come out showing it in one context, and that's extremely satisfying. When I was at Harvard as an undergraduate, it was a very heady experience for me. I felt, God, universities are not simply about teaching, they're about creating new knowledge, new ways of thinking. Isn't that exciting? I someday want to participate in that kind of an environment. And when I came to Columbia, I found that environment. This is a fantastic place. It has a great tradition for scholarship. And I have really taken advantage of that in every possible way. To begin with, I've always enjoyed teaching, but I took it more seriously here. We organized a completely new course that went from molecules to psychoanalysis, literally. And then after several years, I thought maybe we'd put a syllabus together. So each lecture would sort of summarize what they did. And we had a very nice syllabus. And then I thought, gee, why only Columbia? We made a textbook out of it. And Principles of Neuroscience, which this year won an award from the Society of Neuroscience as being, the standard in the field. Moreover, the level of interactions of colleagues here is extraordinary. I would never have gotten the Nobel Prize if I was not here. So one of the things that really energized me, what made it possible for me to clone Krebs, for example, was my collaboration with Richard Axel. He was getting a little tired with what he was doing in molecular biology, and he was looking for new worlds to conquer, and he was thinking about the nervous system. And I wanted to do molecular biology. I knew no molecular biology. So he knew nothing about the brain. I knew nothing about molecular. We started a collaboration together on Aplisia, and I certainly have benefited enormously from not only that collaboration, but our continued friendship. In 1983, the Howard Hughes Medical Institute decided neurobiology is ripe for major investment. They wanted to fund 4 groups throughout the country. That gave us an opportunity with the resources they made available to us to recruit additional people. Tom Jessel, we brought him from Harvard to join us. Steve Siegelbaum, who was here, was about to leave. We encouraged him to stay. So neurobiology, neuroscience became a fantastically rich, interconnected, interesting field that spanned a variety of subtopics here. And over the last decade or so, we've gone from strength to strength. So we are just unbelievable. In all immodesty, I think we're without doubt the most interesting and probably most powerful group in the world. And then we have a president, his name is Lee Bollinger. He actually approached me, I think, initially, and he said, do you think that the neurobiology community would be interested in moving downtown to a new campus that Columbia is going to initiate? And I said, this is clearly not my decision, but let me speak to my colleagues about this. And we discussed it, and we said, this is an opportunity of a lifetime. Can you imagine having a building that is concerned just with brain sciences, in which we can bring engineers, physicists, chemists, psychologists all into the same building? We can bring neuroscience to a completely new level. Because although I've indicated to you the richness of neuroscience, the problems we want to attack, the problems of the human mind, we are decades away from understanding them. We need to amass power. We need the opportunity to interact. We need the environment in which to interact. Of the sciences that are emerging in the 21st century as being really dominant sciences, you've got to say brain science is among them. I mean, I would think it is the most interesting science. So just as, the study of the gene was the most important question 1950 to 2000, so the study of the mind, next 100 years at least. Number one. Number two, he realized that brain sciences could be a bridge between the sciences and humanities. I mean, we spoke about psychology and neuroscience coming together, but what about arts? How one looks at a painting, how one responds to a work of art. We know a lot about perception. We're beginning to understand emotion. We're understanding empathy. We should begin, over the next few decades, to contribute to a dialogue between art historians, artists, and neuroscientists. In area of decision-making, there's a neurobiology of how experimental animals make decisions that bears a lot of similarity to economic theories about decision-making. Sociology, the biology of social interactions, in some cases determined by genes, in some cases determined by hormones. This is something that neurobiologists become very interested in. There's a single gene in the worm that has a slight change, one amino acid flipped from one to another, allows a worm to either go for food alone as an isolate or to go for food as a social group. So you can see that some aspects of behavior, social behavior, are genetically determined. Most, of course, are determined by experience. So even the questions is, how can an individual in Vienna, be civilized one day and be evil the next? These are questions that hopefully someday we will get some insight into. We'll understand that the plasticity of the brain can allow somebody like you with a single nose, 2 eyes, and 2 ears to be capable of the most generous deeds and to be socialized to do terrible things. You know, C.P. Snow spoke about the divide between the humanists and the scientists, that everybody can understand Shakespeare, so the scientists are literate to some degree, but the humanists don't have access to science, and we need to do that. One way to do this is to find areas of common interest and to start a dialogue between this. This is just a fantastic time for doing this. It's very early. These in-between areas are very soft, but they're so fantastically interesting. And can you imagine being an undergraduate at Columbia and seeing these new areas of knowledge emerge? So insofar as universities create new knowledge, Lee Bollinger saw that this is not a single area. This essentially is a university-wide problem. And I think this is why he felt it'd be wonderful to have this as one of the initial things in Manhattanville. Let me just tell you why this is so important for me. You see my office. It's really quite wonderful. The gym is next door. I didn't do it today, but most days I go swimming there between 1 and 2. And my wife works 2 buildings away. She has been Columbia faculty even before I was. So my life here is quite comfortable. Riverdale, where I live, is 10 minutes away by car, and I have a parking space in this building. So I couldn't ask for a better life. But when I think of the future for neuroscience and Columbia, there's no question I'm going to have to travel 10 more minutes, and I'm going to have to find another pool, because I think the science will be so extraordinary, and since this is what drives all of our actions in the long run, it's something we want to do and we have to do. I mentioned before that one of the functions of a university is to create new knowledge, and I've been keenly aware as a refugee, if you will, who's benefited enormously from the American scene, what America has contributed to science per se. And that is, in the 20th century, the United States emerged as the leading nation in science. Why did it do so? I would argue it did so because of Columbia University. And that is Thomas Hunt Morgan, who was the first one to discover that genes are located in a chromosome in the biology department at Columbia, changed the way science is done throughout the world. He introduced democracy, American democracy, into the laboratory. So your significance in a laboratory is not determined by who your father is or how much money you have, but by the nature of your ideas. And he had undergraduate students at Columbia be co-authors of his papers and of his book, because they injected important ideas into science. And this made an enormous impact. And I benefited. Grinfest, you know, treated me in the most wonderful way right here at Columbia, but this spread throughout the country. And throughout the history of Columbia, I mean, you know, cultural anthropology came with Margaret Mead, Boas, all these people. We've introduced many, many new ways of thinking about the world. And I would like to think that out of Columbia will come a new way of thinking about mind and brain, not only enhancing our understanding of the nature of how the brain works, but also fulfilling Lee Bollinger's vision of creating a new kind of knowledge, one that bridges between brain sciences and the humanities. If Columbia achieves that, that's a wonderful testimony to the Bollinger period. One of the things that amazed me being 80 years old is that I get as much joy out of science today. In fact, probably in some ways more, because I don't have the anxiety I had at the beginning of my career about science than I did when I began. And that's because exciting things keep on popping up. One's career evolves, and this has given me an opportunity to do certain things that I've really always wanted to do. So I've been interested in art, and I've been interested in Austrian Expressionist art, and to do a book about art and science has been wonderful. And I mentioned to you I did the autobiography and this documentary, and now I have a chance to do this with Charlie Rose. These are wonderful things. And as long as I continue to get pleasure out of this, I just want to keep on doing it as long as I possibly can.